Right now, somewhere in the skin of your forearm, a cell has just finished copying its entire genetic library. All 6 feet of DNA coiled into a nucleus smaller than a speck of dust, and it is checking its work. Enzymes move along the new strand like proofreaders under a desk lamp, comparing letter to letter, catching mismatches, snipping out the wrong ones, and pasting in the correct bases before the cell allows itself to divide. This happens billions of times a day across your body without your awareness, the way a city's water mains get patched at 3 in the morning while everyone sleeps through the sound of the trucks. In your bone marrow tonight, stem cells are producing roughly 2 million new red blood cells every second, each one squeezing out its own nucleus so it can fold thin enough to slip through capillaries no wider than a hair. In your gut lining, cells that were born 5 days ago are already being shed and replaced, the entire surface renewing itself faster than your bed sheets get washed. Even as you lie still, listening to this, your body is running one of the most elaborate maintenance operations in the known universe, and it has been running continuously since before you could form a memory. It's worth sitting with that image for a moment, because it changes the shape of the question we're about to spend a long, quiet while exploring. We tend to think of growing older as a slow fading, like a photograph left in sunlight, colors leaching out year after year, until only a pale outline remains. But that image is misleading, and it's misleading in a specific way that matters. Fading implies passivity, a picture doing nothing while the world acts upon it. What actually happens inside a living body is closer to the opposite. Your cells are not passively fading. They are working, constantly, to keep you intact, and much of what we call growing older turns out to be less about decline and more about the slow accumulation of small compromises in a system that is trying, with real effort, not to compromise at all. Think about a stone bridge, one of the old ones, the kind built from cut granite blocks fitted together without mortar, relying on compression and gravity to hold the whole structure up. The Pont du Gard in southern France has stood for roughly 2,000 years, and it hasn't stood by doing nothing. Water has seeped into hairline cracks and frozen and expanded them. Wind has worn at exposed edges. Moss has crept into joints, and somewhere in that span of centuries, humans came along more than once to reset stones, repoint gaps, and shore up sections that had begun to lean. The bridge that stands today is not the bridge that was laid 2 millennia ago. It is a bridge that has been continuously repaired, continuously renegotiated with time, and it still shows wear, because repair is never quite the same as prevention, and prevention is never quite the same as permanence. A body is something like that. It is not simply eroding, it is being rebuilt, over and over, by processes so intricate that biologists are still mapping their full extent, and what we call aging is, in large part, the story of what happens when rebuilding starts to fall slightly, imperceptibly, behind the pace of wear. This is a strange thing to realize, because it means aging isn't really a single event or a single mechanism, in the way that, say, a broken bone is a single event with a clear before and after. There is no moment when your body flips a switch and begins to age, the way a light bulb might flip from lit to unlit. Instead, there is a set of overlapping processes, some of them beginning astonishingly early, that shift in relative balance over the course of a lifetime. Cellular repair, happening a little less efficiently here, molecular byproducts, accumulating a little more there. Communication between cells growing slightly noisier, like a radio signal drifting further from its station with each passing mile. These processes don't march in lockstep, and they don't proceed at the same rate in every tissue, or even in every person. A cardiologist and a dermatologist and a neurologist studying the same 70-year-old patient might each describe a different pace of change, because the heart and the skin and the brain are, in a very real sense, aging somewhat independently, shaped by different stresses, different repair capacities, different histories of use. For a long time, the way we talked about growing older reflected this fragmentation. Medicine developed specialty by specialty, organ by organ, and so did our mental picture of aging. We learned about the heart weakening, the joints stiffening, the eyes losing their ability to focus on close objects, a change so common and so predictable that reading glasses have been documented since at least the 13th century, when Italian glassmakers began grinding convex lenses for monks and scholars whose eyesight had begun to blur on the page. Each of these changes got studied in isolation, described in its own textbook chapter, treated by its own specialist, and there's real value in that kind of focused attention. But it can also leave you with the impression that aging is simply a checklist, a tour through the body's organs, in which each stop reveals another item wearing down, unrelated to the others, as if you were walking through a museum of separate exhibits. What researchers have come to understand more recently, particularly over the last few decades, is that many of these seemingly separate declines share common roots. The stiffening in a joint, and the fogging of memory, and the change in how skin heals after a scrape are not entirely separate stories. They are downstream expressions of some of the same underlying cellular processes, playing out differently depending on which tissue they touch. The way the same weather system might bring flooding to a river valley and drought to a hillside a few miles away, both consequences of one shifting pattern, expressed through different terrain. This doesn't mean every kind of decline traces back to one single cause. Biology rarely offers that kind of tidy simplicity, and it would be a disservice to pretend otherwise. But it does mean that thinking about aging organ by organ, as though the heart's story and the brain's story were unconnected, misses something important about how the whole system actually behaves. So instead of touring the body from head to toe, cataloging what tends to go wrong and when, we're going to spend our time together tonight approaching aging as what it actually seems to be, a process, unfolding gradually, built from cellular mechanisms that were, for the vast majority of human history, invisible to us entirely. People noticed the outward signs, of course: gray hair, slower healing, the particular fatigue that settles differently into an older body than a younger one. But the machinery behind those signs remained hidden until relatively recently, tucked away at a scale no unaided eye could reach. It's only in the last century, and especially the last few decades, that scientists gained the tools to actually watch this machinery operate, to see the proofreading enzymes at work, to trace the slow chemical changes that accumulate in a cell over years and decades. That's the story worth telling slowly, without rushing toward conclusions, the way you might listen to rain start outside a window, first a few scattered drops, then a rhythm, then something you stop noticing as separate sounds and start experiencing as one continuous presence. We'll begin, as it happens, with something small, something that sits at the very edge of what a cell can measure about itself, a kind of internal counting mechanism that researchers didn't even suspect existed until surprisingly late in the 20th century. And that changed, almost overnight, how an entire field understood what a cell's lifespan actually meant. But before we follow that thread inside the cell, it's worth pausing on a question that sounds almost too simple to ask out loud. What do we actually mean when we say someone has aged? It seems like it should have an obvious answer. You could just look at a calendar. Someone born in a particular year has lived a particular number of years, And that number climbs steadily, predictably, 1 unit at a time, the same for everyone, everywhere, regardless of how they've spent those years. That's chronological age, and it has the enormous advantage of being easy to measure. You don't need a laboratory. You just need a birth certificate, or a memory, or someone else's memory of the day you were born. But anyone who has spent time around people knows this number, while true, doesn't tell the whole story. 2 people can share a birth year and arrive at 50 in remarkably different conditions. One climbs stairs without noticing them. The other pauses at the landing, catching a breath that didn't used to need catching. One's blood vessels are still supple, still able to widen and narrow the way young vessels do, adjusting smoothly to the demands of a brisk walk or a stressful phone call. The others have grown stiffer, less responsive, more like a garden hose left too long in the sun. Same number of years lived, very different bodies carrying them. This is the gap that researchers point to when they talk about biological age, as distinct from chronological age. And it's a gap that turns out to matter enormously, because it's biological age, not the number on the birthday cake, that seems to track most closely with the things people actually worry about. Risk of illness, risk of frailty, how much time remains, and how much of that time will feel vigorous rather than merely endured. Now, biological age sounds like it should be a single measurable thing, the way chronological age is. A number you could learn by drawing blood or running a scan, the way you'd learn your cholesterol level or your blood pressure. And scientists have tried, for decades, to find exactly that. But here's where things get genuinely interesting, and also genuinely more complicated than the tidy phrase biological age tends to suggest. There is no single dial inside the body that reads out a true biological age the way a car's odometer reads out miles traveled. Instead, there are many different systems, aging at somewhat different rates, responding to somewhat different pressures, each one offering only a partial window into the whole. Think about how you might guess the age of an old house without asking anyone directly. You could look at the exterior paint, flaking in places, sun-bleached in others. That tells you something. But paint gets redone. You could look at the wiring, still original in some houses from decades past, humming along under a layer of dust behind the walls, invisible unless someone opens a panel. You could look at the foundation, which shifts slowly and unevenly depending on the soil beneath it, the drainage around it, the load it's carried over the years. Each of these tells a partial story, and none of them agrees perfectly with the others, because a house doesn't age as one uniform object. It ages as a collection of systems, each with its own materials, its own exposure to weather, its own maintenance history. Ask only about the paint, and you'll underestimate how tired the foundation has become. Ask only about the foundation, and you might miss that the wiring is quietly becoming a hazard. Bodies work something like this, and it's part of why aging research resists easy summary. Researchers have proposed and studied a whole range of biological clocks, ones that read the chemical modifications sitting on top of our DNA, a pattern that shifts in fairly regular ways as cells divide and years pass. Ones that measure how quickly proteins in the blood degrade or accumulate, a kind of chemical weathering. Ones built from something as simple and human as grip strength, or how fast someone can walk a short measured distance, or how efficiently their lungs can move oxygen into blood during exertion. Each of these has been shown, in one study or another, to correlate with future health outcomes better than chronological age alone does. And yet, none of them, not one, captures the whole picture on its own. A person's epigenetic clock might read younger than their calendar age, while their cardiovascular system tells a different story entirely. It isn't that one measurement is right and the others are wrong. It's that aging isn't the kind of process that reduces cleanly to a single number because it isn't happening in a single place. There's another concept worth sitting with here, one that doesn't get talked about as often outside of medical circles, and it's called functional reserve. It's a quietly important idea because it explains something that puzzles a lot of people about how illness seems to behave differently in younger and older bodies. Imagine a young adult catches a bad flu. Their heart rate climbs, their fever spikes, they feel wretched for a few days, and then, in the overwhelming majority of cases, they recover because their organs have capacity well beyond what's needed for ordinary daily living. Healthy young kidneys filter far more blood than strictly necessary. A healthy young heart can, in a pinch, pump several times the blood volume it manages during a quiet afternoon. That extra capacity, that cushion between what the body normally uses and what it's actually capable of producing under strain, is functional reserve. It's a buffer. And like most buffers, it tends to shrink with time, not because any single organ suddenly fails, but because the margins narrow gradually, year after year, often without any noticeable symptom at all, until one day, a stress that would have been trivial in youth, the same flu, a minor infection, a few nights of poor sleep, a hip fracture and the surgery that follows, suddenly reveals how thin the cushion has become. The organ was never broken in any dramatic sense. It simply had less room left to absorb a shock. This is part of why mortality risk, when researchers chart it against age, doesn't rise as a straight line. It curves upward, and it curves upward at an accelerating rate, a relationship first described with real mathematical precision back in 1825 by Benjamin Gompertz, a British actuary working, of all places, for an insurance office, trying to price life insurance policies more accurately. He wasn't a physician. He wasn't studying cells or tissues. He was working with columns of numbers, tables of deaths recorded by age, trying to find a pattern that would let his company set fair premiums. And he found one, a curve that still bears his name, showing that the risk of dying roughly doubles across a fixed number of years, again and again, through much of adult life. It's a strange thing to realize that some of the earliest, most durable insight into the shape of human aging came not from a laboratory but from an insurance ledger, from a mathematician quietly noticing that the buffer everyone carries thins out at a fairly predictable compounding rate as the decades accumulate. So when scientists talk about aging today, they're really juggling several related but distinct ideas at once, and it helps to keep them loosely separate in your mind, the way you might keep several soft threads from tangling. There's the simple passage of time, chronological age, which nobody disputes and nobody can change. There's biological age, the messier, multidimensional reality of how worn or resilient a person's tissues and systems actually are, which doesn't move in lockstep with the calendar and varies meaningfully between two people born on the same day. There's functional reserve, the shrinking cushion between ordinary demand and maximum capacity, which explains why the same illness can be a minor inconvenience in one decade of life and a genuine crisis in another. And there's mortality risk itself, the statistical shadow cast by all of this, rising not steadily, but with a kind of quiet acceleration that a 19th century actuary noticed in his numbers 100 years before anyone understood the biology behind it. None of these, on their own, is aging. They're more like different instruments, each capturing a different note of the same slow, layered sound, none of them sufficient by itself to play the whole piece. And that raises the obvious next question, the one biologists spent much of the 20th century circling without quite being able to answer. If aging isn't one thing happening in one place, where, if anywhere, does it actually begin? It's a question that eventually led researchers down into the cell itself, to a discovery so small and so strange that when it first appeared in the scientific literature, hardly anyone recognized what they were looking at. Before biology could answer where aging begins, it first had to answer a stranger question, one that sounds almost like a riddle. Why does aging happen at all? Not how, not through what molecular pathway, but why evolution, which is generally so ruthlessly efficient at preserving useful traits, would allow something as universally destructive as aging to persist across nearly every branch of the animal kingdom. If natural selection is supposed to favor whatever helps an organism survive and reproduce, then a body that slowly falls apart seems like exactly the kind of flaw selection should have eliminated long ago. And yet here it is, in fruit flies and elephants and roses and men, this slow unwinding that nobody asked for and nothing seems able to prevent. For a long time, this puzzle sat quietly at the edge of biology, more of a philosophical curiosity than a research program, the kind of thing a scientist might wonder about on a long train ride without ever writing down. It was a British immunologist named Peter Medawar who first gave the puzzle a proper shape, in a lecture he delivered at University College London in 1952, a lecture with the plain, almost modest title, An Unsolved Problem of Biology. Medawar was not, at that point, someone anyone would have expected to revolutionize the study of aging. His real expertise was in tissue transplantation and immune rejection, work that would eventually earn him a Nobel Prize for entirely different reasons. But he had a restless, mathematical curiosity, and he approached aging the way an engineer might approach a structure that seemed to fail for no obvious reason, by asking not what was breaking, but what forces had shaped it to break in the first place. His insight, when it came, was almost embarrassingly simple once you saw it, the way the best insights so often are. Natural selection, he pointed out, only has power over traits that affect survival and reproduction before an organism has finished reproducing. Imagine, he said, a population of animals in the wild, constantly exposed to predators, starvation, disease, and accident. Very few of them, statistically, will live long enough to reach old age at all. Most will die young, not from anything resembling aging, but from the ordinary hazards of being alive in a dangerous world, which means that any gene that causes harm only late in life, after most of the reproducing has already been done, is essentially invisible to natural selection. Selection cannot weed out a flaw it never gets the chance to see. It is, Medawar suggested, a little like a landlord who only inspects a rental property during a tenant's first 5 years there. Any damage that only becomes visible after that inspection window closes can accumulate indefinitely, unnoticed and unpunished, simply because nobody with the power to intervene is still looking. Medawar called this idea mutation accumulation, and it explains something true and important, though it leaves something out too, which other scientists would soon notice and try to fill in. It explains why aging exists as a kind of blind spot, a region of biological life that evolution simply never bothered to polish, because in the wild, hardly anything survives long enough to be affected by it. But it treats these late-acting harmful genes as essentially neutral, as junk that slips through unnoticed, rather than as something that might actually be favored, might even be selected for on purpose. And that is where a quieter, in some ways more unsettling idea entered the picture, 5 years later, from an American evolutionary biologist named George Williams. Williams, writing in 1957, suggested something that still has a way of catching people off guard the first time they hear it late at night, turning it over slowly, the way you might turn over a stone in your palm. What if some genes are not just neutral in old age, he asked, but actively harmful there, and yet still get passed down generation after generation because those very same genes are helpful earlier in life, when it actually matters to survival and reproduction? He called this antagonistic pleiotropy, a mouthful of a phrase for a fairly intuitive idea. Pleiotropy simply means that a single gene can influence more than one trait, the way a single decision made early in a person's life might ripple outward and touch parts of their future they never anticipated. Antagonistic means these effects can pull in opposite directions, helpful in one season of life, harmful in another. A gene that makes bones grow strong and dense in youth, useful for a body that needs to run and climb and fight, might be the very same gene that, decades later, contributes to the stiffening and brittleness of aging joints. A hormone that sharpens fertility and vigor in early adulthood might, through some entirely different downstream effect, quietly raise the risk of certain cancers much later on. Natural selection, faced with a trade-off like this, will almost always choose the early benefit over the late cost, because in the calculus of survival and reproduction, a young body that thrives today matters far more than an old body that suffers decades from now, especially in a world where most individuals, historically, never lived to experience that later cost at all. It is a strange kind of bargain, struck without anyone's awareness or consent, favoring vigor now over resilience later, again and again, across countless generations, until the bargain becomes written into the very architecture of the body. Together, these two ideas, mutation accumulation and antagonistic pleiotropy, reframed aging entirely. It was no longer a mystery of decline for its own sake, no longer a design flaw waiting to be discovered and fixed, but something closer to an inevitable shadow cast by the shape of natural selection itself, a shadow that falls precisely where selection's light doesn't reach. Aging, in this view, isn't something evolution built. It's something evolution simply failed to prevent, and in some cases, quietly encouraged, as a side effect of building something else. There was a third piece still missing, though, a question about resources rather than genes, and it came from a British biologist named Tom Kirkwood, working alongside his colleague Robin Holliday in the late 1970s. Kirkwood asked a question that feels almost domestic in its simplicity. Every living body, he pointed out, has to divide a finite budget of energy between two competing projects. One project is maintenance, the continual, unglamorous work of repairing damaged cells, correcting errors in DNA, replacing worn-out proteins, keeping the whole intricate machine running smoothly. The other project is reproduction, the energetically expensive business of producing offspring and, in many species, caring for them afterward. No organism has infinite resources for both. Every calorie spent on repair is a calorie not spent on reproduction, and every calorie spent on reproduction is a calorie not spent on repair. Kirkwood called this the disposable soma theory, borrowing the word soma, which simply means the body, as distinct from the germline, the reproductive cells that carry genetic information into the next generation. And his central image is worth sitting with for a moment, because it's a genuinely useful way to picture the whole strange bargain of being alive. The germline, in a sense, is designed to be immortal, or at least renewable, carried forward indefinitely from parent to child to grandchild. The soma, the body itself, the hands and lungs and skin, is comparatively disposable, built well enough to survive long enough to reproduce successfully, but not engineered for indefinite upkeep beyond that, because indefinite upkeep would come at too steep a cost to reproduction itself, and evolution, again, does not reward indefinite upkeep. It rewards successful reproduction and whatever level of bodily maintenance happens to be sufficient to achieve it. What all three of these ideas share, mutation accumulation, antagonistic pleiotropy, and disposable soma, is that they answer the question why without ever really touching the question how. They explain, at the level of populations and generations and evolutionary trade-offs, why a body would be built to eventually fail rather than persist forever. But they say almost nothing about what is actually happening inside a single cell, in a single body, on a single ordinary afternoon, as that failure slowly, quietly unfolds. That is a separate question entirely, a question of mechanism rather than motive, and it would take biology several more decades and a series of accidental, almost overlooked laboratory observations to even begin answering it. So let's start there, with mechanism, with the actual physical texture of what goes wrong inside a cell, because the story turns out to be stranger and more interesting than the word wear suggests. The wear and tear metaphor is old, older than modern biology itself, and it has a certain intuitive appeal that's hard to shake. A body is like a machine, the thinking goes, and machines wear out. Gears grind down, metal fatigues, rubber cracks with age and sunlight. Run anything long enough and the friction of simply operating will eventually degrade it, and surely bodies are no different. Cells rubbing against the business of living until something gives way. It's a comforting idea because it's so familiar, and it isn't entirely wrong, but it's incomplete in a way that matters, because a body is not actually much like a machine. A machine doesn't repair itself. A body does, constantly, almost unbelievably so, and understanding aging means understanding not just the damage but the repair, and more specifically, why the repair, however remarkable, is never quite complete. Consider the DNA inside a single one of your cells right now, coiled and folded into a nucleus so small it would take roughly 10,000 of them lined up to span the width of a human hair. That DNA is not sitting quietly. It is being read, copied, unwound, rewound, and physically jostled by the ordinary molecular traffic of a working cell, thousands of times a day, in every one of the trillions of cells that make up a body. And in the course of all that activity, damage happens. Not occasionally, not as some rare misfortune, but constantly, as an ordinary and unavoidable feature of being made of chemistry that runs at body temperature in a wet, crowded, chemically active environment. Estimates vary depending on cell type and method of counting, but a single human cell may sustain something on the order of tens of thousands of DNA lesions in the course of a single day. A break in a strand. A base chemically altered so it can no longer be read correctly. A place where two strands have become improperly fused together. These are not exotic events caused by dramatic exposures, though certainly radiation and certain chemicals can add to the tally. Mostly, they are simply the byproduct of the cell doing what a cell does, of metabolism itself. Oxidative chemistry deserves particular attention here, because it's one of the more historically important threads in this story. In 1954, a chemist named Denham Harmon was sitting at his desk at the University of California, thinking, by his own account, about a completely unrelated problem, when an idea occurred to him that he later said arrived almost fully formed. He'd been reading about radiation damage and about a class of highly reactive molecules called free radicals, and it struck him that the body's own ordinary metabolism, the very process of converting food and oxygen into usable energy, might be quietly generating similar damage all the time, over the entire course of a life, without any radiation exposure required at all. Free radicals, and more broadly what are now called reactive oxygen species, are molecules with an unpaired electron, which makes them chemically unstable and eager to react with whatever they encounter, stealing electrons from nearby molecules to settle themselves, and in doing so, damaging those molecules in turn. They are produced, in small but steady quantities, inside the mitochondria, the structures within cells that generate energy by processing oxygen, which means that the very act of breathing, of extracting usable energy from the air, carries with it a small, continuous cost. Harman's free radical theory of aging, refined over the following decades and eventually folded into a broader idea sometimes called the oxidative damage theory, proposed that this slow chemical erosion, this steady background hum of molecular damage from ordinary respiration, was a significant driver of the aging process. Proteins get oxidized and lose their proper shape. Fats in cell membranes get oxidized and become brittle or leaky. DNA itself is a frequent target, and one of the most common products of this oxidative damage is a molecule with a name you don't need to remember, but which researchers use almost like a chemical fingerprint, a marker they can measure in tissue to estimate how much oxidative wear has accumulated over time. It's worth saying clearly that the free radical theory, in its strongest original form, hasn't held up perfectly under decades of further testing. Experiments where researchers boosted animals' levels of antioxidant defenses, expecting this to slow aging and extend lifespan if oxidative damage really were the central driver, often produced disappointing or mixed results, and some studies even found that certain reactive oxygen species, in modest amounts, seem to serve as useful signals within cells rather than pure agents of destruction, part of the ordinary language cells use to communicate stress and adjust their behavior. So the picture is more complicated than Harman's original elegant idea, as pictures in biology so often turn out to be. But the underlying observation that ordinary metabolism generates chemical damage as a byproduct, and that this damage accumulates over a lifetime unless something intervenes, remains an important piece of the larger puzzle, one thread among several rather than the single explanation researchers once hoped it might be. Now, here is where the story becomes genuinely interesting, and where it moves past a simple wear metaphor, because cells are not passive recipients of all this damage. They have, over billions of years of evolution, developed an extraordinarily sophisticated set of repair systems, whole suites of specialized proteins whose entire function is to patrol the genome, detect exactly this kind of lesion, and fix it. There are enzymes that recognize a mismatched or damaged base and excise it, cutting it out like a proofreader circling a typo, then filling in the correct letter using the opposite strand as a template. There are separate systems for repairing double-strand breaks, for correcting errors that slip past the cell's own copying machinery, for untangling the kind of damage caused by ultraviolet light or certain chemicals. The scale of this repair effort is honestly difficult to hold in your mind properly. A single human cell may correct the vast majority, something like nearly all, of the tens of thousands of lesions it accumulates each day. That is a staggering amount of ongoing maintenance, performed silently, continuously, in every cell in your body, right now, while you rest. And this is exactly why the wear and tear metaphor falls short. A worn gear doesn't get quietly remachined back to its original shape overnight, but your DNA more or less does, over and over, for decades. The honest and more precise way to think about aging, at this molecular level, isn't simply damage accumulating like dust on a shelf. It's closer to a very slight, very persistent imbalance between two enormous opposing processes: damage generation on one side, repair on the other, both running at tremendous speed and scale, with repair winning the overwhelming majority of individual battles. The lesions that matter for aging are the rare exceptions, the small fraction that slip through uncorrected, or that get repaired slightly wrong, introducing a genuine mutation, a permanent change to the genetic sequence rather than a temporary lesion. Multiply that small fraction by trillions of cells and tens of thousands of daily events and decades of accumulated time, and even a very high repair success rate leaves behind a slowly growing residue of permanent errors. This is also where molecular turnover enters the picture, because DNA isn't the only thing in a cell that degrades and gets fixed or replaced. Proteins, the actual working machinery of the cell, the enzymes and structural components and signaling molecules that do virtually everything a cell needs done, are also being damaged constantly by oxidation, by misfolding, by simple chemical instability, and cells have their own separate systems for detecting and discarding proteins that have gone wrong, breaking them down, and rebuilding fresh copies from scratch. Most proteins in your body today are not the same physical molecules that were there a few weeks ago. They've been replaced, recycled, rebuilt, in a continuous churn that never really stops. Which raises a quieter, stranger question, one worth letting settle before we go any further into how this whole system was actually discovered. The question is simply this: if proteins get rebuilt and DNA gets repaired, what exactly is aging at the level of a single cell when so much of the cell seems to be renewing itself all the time? Part of the answer lives in a structure so small and so quietly important that it went unnoticed by biology for most of the 20th century, tucked away at the very tips of your chromosomes, doing a job nobody had thought to look for. Picture a shoelace. Not the fabric of the lace itself, but the small plastic or metal cap at each end, the aglet, the thing that keeps the woven threads from unraveling every time you tug at it. Chromosomes, the long threads of DNA coiled up inside the nucleus of nearly every cell in your body, have something functionally similar at their ends, called telomeres. They are repetitive stretches of DNA, the same short sequence of genetic letters repeated over and over, thousands of times, not coding for any protein, not doing the kind of active informational work that the rest of the genome does. Their job is structural, protective, a buffer zone. Why would a chromosome need a buffer zone at all? The answer has to do with a peculiar and somewhat inconvenient feature of how DNA gets copied. Every time a cell divides, it has to duplicate its entire genome first, so that each of the two resulting daughter cells gets a complete set. The machinery that does this copying, a cluster of enzymes built around something called DNA polymerase, is extraordinarily good at what it does, but it has one structural quirk. It can only add new genetic letters onto an existing strand by building in one direction, and it needs a short starter sequence, a kind of primer, to get going, the same way you might need a bit of existing scaffolding before you can start stacking bricks on top of it. Along most of the chromosome, this isn't a problem, because there's always more sequence downstream to serve as that scaffold. But at the very last stretch, at the tip, there's no more room. The primer itself occupies real estate that never gets filled back in. So with each round of copying, each division, the very end of the chromosome gets very slightly shorter. Not because anything is being damaged or degraded in the way we usually think of damage, simply because the copying machine runs out of room to finish the job. This is called the end replication problem, and it was worked out independently and almost simultaneously by a Russian theoretical biologist named Alexei Olovnikov and by James Watson, one half of the pair credited with the double helix structure itself, in the early 1970s. Watson noticed the mechanical issue from thinking carefully about how the polymerase enzyme actually behaves. Olovnikov went further and made a genuinely striking leap. He proposed that this small, unavoidable shortening, happening a tiny bit at a time with every division, might function as a kind of built-in counter, a way for a cell to keep track, in a very rough sense, of how many times it had already divided. And if that counter ever ran down far enough, he suggested, it might act as a signal, a stop sign, telling the cell that it had reached some kind of limit. That idea connected neatly to something else that had already been observed a decade earlier, in 1961, by an American anatomist named Leonard Hayflick. Hayflick had been growing human cells in laboratory dishes, feeding them, giving them room to divide, watching them multiply generation after generation, and he noticed something that contradicted the assumption at the time, which was that cells in a dish, given enough nutrients and space, would simply keep dividing forever, the way a lot of people still imagine biological life ought to work if nothing goes wrong. Instead, Hayflick's cells divided a limited number of times, somewhere in the range of 40 to 60 divisions, depending on the cell type and the person the cells came from. And then they stopped. Not by dying. They simply stopped dividing, sat there, remained metabolically active, changed their shape somewhat, and settled into a kind of permanent standstill. This limit came to be called the Hayflick limit, and for years its cause was a mystery. Olovnikov's telomere idea offered a mechanism, and in the 1980s, that mechanism got direct experimental support, largely through the work of Elizabeth Blackburn, working initially with a peculiar single-celled pond organism called Tetrahymena, along with Carol Greider and Jack Szostak. Together, this research identified telomerase, an enzyme that can actually rebuild those repetitive N sequences, adding length back onto the very tips of the chromosomes. Here is where the story gets genuinely interesting, rather than simply mechanical, because telomerase is not switched on everywhere. Most of the ordinary cells in your body, your skin cells, your liver cells, the cells lining your gut, keep telomerase largely turned off or active at only very low levels, which means, in most of your tissues, the shortening is allowed to continue, division after division, until eventually some cells reach that Hayflick limit and stop. This might sound like bad news, like one more slow demolition, but researchers came to understand it rather differently, as something closer to a safety feature. Cancer, at its core, is a disease of cells that have lost the normal limits on their own division, that keep copying themselves indefinitely, ignoring the signals that would normally tell an ordinary cell to stop. A cell with unlimited replicative capacity is, in a sense, exactly what a cancer needs to become dangerous. So a built-in counter, a mechanism that eventually says, enough, you've divided as many times as we're going to allow, functions as a kind of brake on runaway growth. It's telling that the vast majority of cancer cells, when researchers examine them, have found some way to reactivate telomerase, or some equivalent workaround, specifically so they can bypass this limit and keep dividing without end. Evolution, in other words, seems to have arrived at a genuine trade-off. Keep telomerase mostly switched off in most tissues, except that individual cells will eventually retire from the dividing pool, and in exchange gain meaningful protection against one of the more common and most dangerous things that can go wrong in a multicellular body. Long life for the whole organism, purchased partly at the cost of finite life for many of its individual cells. Now, what happens to a cell once it actually reaches that stopping point matters enormously. And this is where the idea of cellular senescence comes in. A senescent cell isn't dead. It's alive, present, still metabolically active, but it has permanently exited the cycle of division and it starts behaving differently than it did before. Notably, senescent cells begin secreting a distinctive mixture of signaling molecules out into their surrounding tissue, inflammatory proteins, growth factors, enzymes that can remodel nearby tissue structure. Researchers gave this secretion pattern a name, the senescence-associated secretory phenotype. And in modest amounts, over a short time, it may actually serve a useful purpose, helping call in immune cells to clear out damaged tissue, or assisting in wound healing. But senescent cells don't always get cleared away promptly. They can linger for months, for years, continuing to secrete that same inflammatory signal into their local environment, disrupting the normal signaling of nearby healthy cells, and as they slowly accumulate across a lifetime, particularly in older tissue, they've been connected to a wide range of the same age-related conditions that show up throughout this whole subject, from joint stiffness to blood vessel changes to certain patterns of tissue fibrosis. Given everything you've just heard, it might be tempting to reach for a clean, satisfying conclusion here, that telomere length is essentially a biological clock, a countdown timer sitting inside each of us, ticking down division by division toward some fixed endpoint. It's an appealing idea. It's also, according to the evidence gathered over the decades since Blackburn and Grider's original discoveries, considerably more complicated and more interesting than that. Telomere length varies enormously between different tissues in the very same body, since some tissues divide constantly, while others barely divide at all across an entire lifetime. It varies between different people of identical age, shaped by inherited genetic differences, by early developmental conditions, by stress and inflammation. Measuring the average telomere length in a simple blood sample turns out to correlate only loosely with someone's actual biological condition, loosely enough that using it as a standalone marker of how fast someone is aging has proven far less reliable than early researchers had hoped, which leaves an open, still, gently unresolved question sitting in the room. If telomere shortening isn't the master clock after all, just one contributing thread among several, what else is actually setting the pace? One place scientists turn to look for that pace setter is not the length of the DNA strand itself, but the pattern of tiny chemical marks sitting on top of it, a layer of information that doesn't change the genetic sequence at all, but changes how that sequence gets read. This layer has a name, epigenetics, coined back in 1942 by a British biologist named Conrad Waddington, who was trying to describe something that had puzzled him for years. Every cell in your body, from a neuron in your brain to a skin cell on your fingertip to a cell lining your gut, carries the exact same DNA, the same full instruction manual, cover to cover. And yet these cells look nothing alike and do entirely different jobs. A neuron doesn't secrete stomach acid. A skin cell doesn't fire electrical signals down a foot-long fiber. If they're all reading from an identical manual, something else has to be determining which pages actually get opened and which stay closed. That something else turns out to involve small molecular tags, the most well-studied of which is called a methyl group, a tiny cluster of one carbon atom and 3 hydrogen atoms, chemically modest, almost trivial-looking on paper. But when one of these methyl groups attaches itself to a particular spot along the DNA strand, usually at a location where a molecule called cytosine sits next to one called guanine, it can act like a small piece of tape pressed over a paragraph of text. The instructions underneath are still there, still perfectly intact, but the cell's reading machinery slides right past that stretch without engaging it. Layer enough of these methyl tags over the genes for, say, producing stomach acid, and a developing brain cell effectively silences that whole section of its own manual, while leaving wide open the sections it actually needs, the ones for building synapses and neurotransmitters. Multiply this process across thousands of genes, and you start to see how one single set of instructions can produce hundreds of wildly different cell types, each one just reading a different, carefully curated combination of chapters. What makes this relevant to aging is a discovery that emerged gradually over the early 2000s and then crystallized rather dramatically in 2013, when a biostatistician named Steve Horvath, working at Ookla, published a piece of work that had taken him years of painstaking, largely unglamorous data analysis to complete. Horvath gathered methylation data from thousands of tissue samples, spanning nearly every part of the body, from people whose ages were already known, and he set out to see whether these methylation patterns, that pattern of tape covering different paragraphs across the genome, changed in any kind of orderly, predictable way as people got older. It turned out they did, remarkably so. At several hundred specific locations along the genome, the degree of methylation crept up or down in a fashion so consistent across different people that Horvath was able to build a mathematical model, essentially a formula, that could take a sample of someone's cells, measure the methylation at those specific sites, and estimate that person's age. Not their age in the sense of when they were born, but something Horvath and later researchers began calling biological age, an estimate that in most people tracks fairly close to their actual calendar age, but that in some people runs noticeably ahead and in others noticeably behind. This tool became known, informally, as an epigenetic clock, and it's worth sitting with why that name is both apt and slightly misleading at the same time. It is a genuine clock in the sense that it keeps remarkably steady time. Feed it a tissue sample from almost any part of the body, skin, blood, saliva, even certain internal organs, and the formula gives you a strikingly consistent estimate. It has proven robust enough and predictive enough that it now shows up regularly in aging research as one of the more reliable available proxies for how someone is aging biologically, correlating with things like cardiovascular disease risk, cognitive decline, and yes, even overall mortality, somewhat better than calendar age alone does in certain studies. Someone whose epigenetic clock reads several years older than their actual age tends to, on average across large populations, face a modestly elevated risk of a number of these age-related conditions down the road. But here is the place where it becomes important to slow down and be careful, because a clock that reliably tells you what time it is on the wall is not automatically the same thing as a mechanism that is causing the hour to change. Horvath's clock, and the many variations of it that other researchers have built since, using slightly different sets of methylation sites, tuned to predict slightly different outcomes, are what scientists call biomarkers. They are readings. Extremely useful readings. Readings good enough to help researchers evaluate whether some new intervention, a drug, a fasting protocol, an exercise regimen, appears to be slowing biological aging in a clinical trial, without having to wait decades to see who lives longer. But a biomarker, no matter how precise, doesn't by itself explain why the methylation pattern shifts in the first place, or whether adjusting that pattern directly would actually change how a person ages, or whether the clock is simply a faithful bystander, quietly recording changes that some other, still poorly understood process is actually driving underneath it. It's a bit like watching the hands of a clock tower move and concluding you understand gravity, when really, you've just gotten very good at reading the hands. This distinction became especially vivid and especially exciting to a lot of researchers, following a set of experiments that trace back to work published by a Japanese scientist named Shinya Yamanaka in 2006. Yamanaka had discovered that if you take an ordinary, fully mature adult cell, something like a skin cell, that has already gone through its whole developmental journey and settled permanently into being skin, and you expose it to just 4 specific proteins, later nicknamed the Yamanaka factors, you can coax that cell to essentially forget what it is. It reverts, over a period of days, back toward something resembling an early embryonic state, capable once again of becoming almost any cell type in the body. The methylation pattern, that carefully arranged tape covering different chapters of the manual, gets substantially wiped and reset in the process. Yamanaka received the Nobel Prize for this discovery in 2012, and it opened an entirely new avenue of aging research, because if methylation patterns can be reset, at least in a laboratory dish, what happens to the other hallmarks of aging when you do that? Some intriguing, still fairly early findings have come out of pursuing that question. Researchers, including David Sinclair's lab at Harvard, have shown that applying a partial, carefully limited dose of those same reprogramming factors to aged mice, just enough to nudge the methylation pattern backward without fully erasing the cell's identity, can in some cases restore youthful characteristics to specific tissues, improving vision in mice with damaged optic nerves, for instance, in work published in 2020. These are genuinely striking results, and they've generated real excitement in the field. But it's worth holding on to some caution here, too, the same caution that applies to the clock itself. These experiments remain in early stages, largely confined to animal models and specific tissues, and the leap from restoring optic nerve function in a mouse to safely reversing aging broadly in a human body is an enormous one, full of unresolved questions about dosing, about cancer risk, given that unchecked cellular reprogramming is uncomfortably close to how certain tumors behave, and about whether what works in a controlled laboratory setting will translate at all to the full, tangled complexity of an aging human system. So where does that leave the search for what's actually setting the pace of aging? The methylation clock gives us one of the clearest windows yet into how orderly and how measurable the aging process really is underneath the surface. But a window is not the same as a door. It shows the pattern changing. It doesn't, on its own, tell us what's turning the pages. And that question, of what mechanism actually drives the shift, pulls us naturally toward another structure inside the cell, one far older in evolutionary terms than the nucleus itself, a set of tiny power-generating structures that carry their own separate, much smaller strand of DNA, and that some researchers have come to see as carrying a story about aging all their own. Mitochondria are strange little residents of our cells, and their strangeness is part of what makes them so interesting to aging researchers. Billions of years ago, the story goes, a larger cell engulfed a smaller bacterium and, rather than digesting it, struck up a kind of permanent arrangement, a merger so complete and so ancient that we still carry the evidence of it in every cell of our bodies. That engulfed bacterium became the mitochondrion, the structure that takes in oxygen and the sugars and fats we eat and converts them into a usable form of chemical energy, a molecule called ATP, which powers essentially everything a cell does, from firing a neuron to contracting a muscle fiber to repairing a strand of damaged DNA. And because of that ancient origin, mitochondria still carry their own small loop of DNA, separate from the DNA in the nucleus, inherited only from the mother, and copied by a slightly different, slightly less careful set of enzymes than the ones that copy the DNA in the rest of the cell. For a long stretch of the 20th century, a researcher named Denham Harman, working in the 1950s, proposed that this arrangement was where aging fundamentally came from. His idea, known as the free radical theory of aging, suggested that the very process of generating energy in the mitochondria produces, as a kind of exhaust, unstable molecules called reactive oxygen species, and that over decades, these molecules accumulate damage in the mitochondrial DNA and in the surrounding cellular machinery, gradually degrading the cell's ability to produce energy efficiently, which in turn degrades the tissue, which in turn degrades the whole organism. It was an elegant idea, and for a long time it was close to the dominant explanation for why we age at all. But subsequent decades of research have complicated it considerably. Experiments where researchers loaded up mice with extra antioxidants, substances meant to soak up those reactive molecules before they could do damage, mostly failed to extend lifespan in any meaningful way. Some experiments even found that increasing oxidative stress under certain controlled conditions made worms live longer rather than shorter, which is close to the opposite of what the simple version of the theory would predict. So the free radical theory hasn't been discarded exactly, but it's been folded into something more complicated. A recognition that mitochondrial function matters enormously to aging, but not necessarily through the straightforward wear and tear story that seemed so intuitive at first. What's emerged instead, and what a great deal of aging research has turned its attention toward in recent decades, is the idea that mitochondria matter less as a source of accumulating damage and more as a kind of sensor, a structure deeply wired into how the cell senses and responds to the availability of food. And that observation connects to one of the oldest and most consistently reproduced findings in the entire science of aging, a finding that predates almost everything else we've talked about. Back in 1935, two researchers at Cornell, Clive McKay and Mary Crowell, published a study showing that rats fed a significantly reduced calorie diet while still receiving adequate vitamins and minerals, lived substantially longer than rats fed freely, not slightly longer. In some of their experiments, dramatically longer, alongside a reduction in tumors and other diseases of old age. It was a startling result for its time, and it has been replicated, in various forms, across an enormous range of species since then. Yeast, worms, flies, mice, and in more limited but still notable ways, primates, all seem to show some version of the same pattern. Eat less, live longer. Not starve, which is a different and dangerous thing entirely, but eat measurably less than you would if food were unlimited and appetite were the only guide. The question that has occupied researchers for the better part of a century since McKay and Crowell's experiment is why. Why would eating less extend life? And the answer that has slowly come into focus involves a set of molecular pathways that function almost like a thermostat for the body's relationship to food, sensing how much fuel is available and adjusting the cell's priorities accordingly. One of the central players in this system is a signaling molecule called mTOR, which stands for Mechanistic Target of Rapamycin, a name that traces back to rapamycin itself, a compound first isolated in the 1970s from a soil bacterium found on Easter Island, of all places, where it was initially studied as an antifungal agent before researchers discovered it had powerful effects on cell growth and, eventually, on lifespan in laboratory animals. mTOR functions as a kind of growth switch inside the cell. When nutrients are abundant, when there's plenty of glucose and amino acids circulating, mTOR activity rises, and the cell shifts its resources toward growth, toward building new proteins, toward dividing and expanding. This makes sense as a strategy when food is plentiful. But that same growth-oriented state, it turns out, comes with a cost, because a cell that is busy building things is a cell that is devoting less energy to cleaning things up, to the ongoing maintenance and quality control that keeps its internal machinery in good working order. That maintenance work has its own name, autophagy, from the Greek for self-eating, a process by which the cell identifies its own damaged or worn-out components, proteins that have folded incorrectly, mitochondria that are no longer functioning well, and breaks them down, recycling their parts into raw material for new construction. It's not unlike the routine a careful homeowner might follow, going through the house periodically and disposing of things that have broken or spoiled, rather than letting clutter accumulate indefinitely. Autophagy runs constantly in the background of a healthy cell, but its activity is closely tied to nutrient signaling, and in particular, it tends to be suppressed when mTOR activity is high, and unleashed when mTOR activity drops. Yoshinori Osumi, a Japanese cell biologist, spent much of his career at the Tokyo Institute of Technology mapping out the genetics of this process in yeast, identifying many of the genes involved, work that eventually earned him the Nobel Prize in Physiology or Medicine in 2016. His research, along with decades of subsequent work in other labs, helped establish that when food is scarce, cells respond not just by conserving energy passively, but by actively ramping up this internal cleanup and recycling process, as though sensing that in a period of want, it makes more sense to refurbish what already exists than to keep building anew. There's a companion system to this as well, an enzyme called AMP, which stands for AMP activated protein kinase, that functions almost as the opposite signal to mTOR, activating specifically when the cell's energy reserves run low, when the ratio of spent to unspent energy currency shifts in a way that signals scarcity. AMP, once activated, tends to suppress mTOR further and to promote autophagy and other conservation measures, and it also influences mitochondria directly, encouraging the cell to build new, more efficient mitochondria, and to clear out the old, poorly functioning ones through a related process, sometimes called mitophagy. Together, mTOR and AMP and the broader network of insulin and insulin-like growth factor signaling that surrounds them form something like a coordinated response team, constantly reading the nutritional state of the body and adjusting a whole cascade of downstream processes: growth versus repair, division versus maintenance, expansion versus cleanup, based on what they detect. Caloric restriction, in this framework, seems to work by nudging this entire system toward the maintenance side of the balance more of the time, dialing down growth signaling and dialing up the cell's internal quality control, and doing so consistently enough over years and years that the cumulative effect on tissue health becomes substantial. It's a genuinely compelling piece of the aging story, one of the most robustly reproduced findings across the whole field, and it has motivated researchers to look for compounds that might mimic these effects without requiring someone to actually restrict their food intake for decades. Drugs like rapamycin itself, or metformin, a diabetes medication that also happens to activate AMP, both of which are currently the subject of ongoing human trials. But it's worth pausing here, gently, on a note of caution, because this is exactly the kind of place where animal research and human application can come apart in ways that are easy to overlook if you're only half listening. A mouse lives 2 or 3 years under laboratory conditions that are nothing like the conditions a human lives under, and a finding that holds beautifully in a short-lived, genetically uniform, carefully controlled rodent population doesn't automatically carry over to a long-lived, genetically diverse species living in an uncontrolled world, and that gap between the mouse cage and the human life is one we'll need to sit with more carefully. We'll return to that gap in a little while, because it matters for almost everything this particular story touches. But for now, let's set the nutrient-sensing network aside and turn to a different, closely related layer of the aging puzzle, one that has less to do with what a cell decides to do with the fuel it has, and more to do with how well it manages the objects it's already built. Because a cell isn't just a factory that produces things, it's also, constantly, a kind of housekeeper, and like any household, it accumulates clutter, and the ease or difficulty with which it clears that clutter turns out to be another one of the threads running through the whole aging story. Think for a moment about proteins themselves, not as abstract biological currency, but as physical objects, because that's what they are. A protein starts its life as a long, floppy chain of amino acids, strung together one after another according to instructions read out from DNA, and in that raw, unfolded state it's essentially useless. It has to fold, it has to collapse and twist, and coil itself into a specific three-dimensional shape, the way a long strip of paper might be folded into an origami crane, and it's that shape, that precise geometry, that gives the protein its function. A folded enzyme has pockets and grooves shaped just right to bind its target. A folded structural protein has surfaces shaped just right to link up with its neighbors. Change the shape and you change, or destroy, the function. The same way crumpling that paper crane back into a ball leaves you with something that no longer resembles a bird, no matter how much paper is still technically there. For a long time, biologists assumed that folding was mostly automatic, that a protein chain would simply find its correct shape on its own, guided by nothing more than the basic chemistry of its amino acids seeking their most comfortable arrangement. And there's real truth in that idea. The American biochemist Christian Anfinsen showed in the 1950s and 60s that a small protein, unfolded in a test tube, could refold itself correctly, with no other help at all, a discovery that eventually earned him a share of the Nobel Prize. But inside a living cell, folding turns out to be a much more crowded, much more hazardous process than it is in the clean isolation of a laboratory beaker. Cells are dense, jam-packed environments, closer to rush hour on a train platform than to open water, and a chain of amino acids trying to fold correctly in that crush is constantly at risk of colliding with its neighbors, sticking to the wrong surfaces, getting tangled into some misshapen dead end instead of its proper form. This is where a remarkable class of proteins called chaperones comes in, and the name is a good one, because that's essentially their job. Chaperones don't provide the final shape of a protein themselves. They don't dictate what a young protein should become. They simply create the conditions, sometimes literally enclosing a newly forming protein inside a small protected chamber, that let the folding happen safely, shielded from the crowd, until the correct shape is found. Some chaperones also do something more like triage, recognizing proteins that have folded incorrectly, or that have come unfolded through heat or stress or ordinary wear, and either giving them a second chance to refold properly, or, if that fails, tagging them for removal. This tagging system itself was a major discovery, worked out in detail by Aaron Ciechanover, Avram Hershko, and Erwin Rose, who found that cells attach a small molecular flag, called ubiquitin, to proteins destined for destruction, work that also earned a Nobel Prize, this one in 2004, more than 4 decades after Anfinsen's. Once a protein is flagged this way, it's typically fed into a barrel-shaped structure called the proteasome, which unfolds it and chops it into fragments, recycling the raw materials for future use. But proteins aren't the only thing that needs periodic destruction and renewal. Whole damaged structures, clumps of misfolded material, too large or too tangled for the proteasome to handle, worn-out organelles, even invading microbes, are handled by a different system entirely, one built around small membrane-bound sacs called lysosomes, essentially the cell's stomach, filled with a cocktail of enzymes capable of breaking down almost anything unlucky enough to end up inside them. The process of delivering material to the lysosome for this kind of bulk demolition is called autophagy, from Greek roots meaning, roughly, self-eating, a term coined in the 1960s by the Belgian scientist Christian de Duve, who had discovered the lysosome itself a decade earlier and who would go on to share a Nobel Prize of his own for that work. Autophagy sounds almost violent when you say it plainly, a cell eating itself, but in practice it's closer to composting, a way of breaking down what's no longer useful into pieces, simple enough to be rebuilt into something new. Now, here's where all of this starts to connect back to aging. Each of these systems, the chaperones that guide folding, the ubiquitin tagging system, the proteasome that shreds flagged proteins, the lysosome, and the autophagy machinery that clears out larger debris, works reasonably well on its own, but none of them works with perfect efficiency, and none of them works in isolation from the others. They depend on energy, much of it ultimately supplied by those same mitochondria we were discussing a little while ago. They depend on the cell's overall metabolic state, which is exactly what mTOR and AMP are constantly adjusting. And they depend, simply, on capacity, on there being enough chaperones, enough functioning proteasomes, enough healthy lysosomes to keep pace with the rate at which damaged and misfolded material is produced. When a young, healthy cell is running well, that pace is more or less balanced. Production of clutter roughly matched by the rate of cleanup, the way a well-run household manages to keep the dishes from piling up, even though new dishes get dirtied every single day. But over time, in ways researchers are still working to fully map out, that balance seems to tip. Chaperones become somewhat less efficient. Lysosomes accumulate a strange, indigestible pigment called lipofuscin, sometimes described as cellular debris that even the lysosome's powerful enzymes can't quite break down, which builds up inside these organelles over decades the way sediment slowly narrows a riverbed, leaving less room and less capacity for everything else that needs processing. Autophagy itself, that broader housekeeping process, tends to become less active with age in many tissues that have been studied, which is part of why interventions like caloric restriction, which stimulates autophagy as one of its downstream effects, have drawn so much interest from researchers thinking about these questions. And when cleanup slows down even slightly, while production of damaged material continues at its usual rate, the clutter starts to accumulate. Misfolded proteins that would once have been caught and corrected or destroyed begin to persist a little longer, and in some tissues, particularly the brain, they can begin to clump together into the kinds of aggregates associated with certain neurodegenerative conditions, though the exact relationship between this general decline in proteostasis and any specific disease remains an active and, in places, contested area of research. It isn't only what's happening inside cells that shifts with age either. The material between cells changes too, something worth mentioning because it's easy to forget, tucked away as it is outside the tidy boundary of the cell membrane. This space is filled with a scaffold called the extracellular matrix, a mesh of proteins like collagen and elastin that gives tissues their structure, their elasticity, the particular give and bounce of young skin, or the flexibility of a young artery. These matrix proteins are also subject to wear, though of a slightly different kind, since they exist for such a long time, sometimes years or decades, that they're more exposed than most proteins ever are to slow chemical reactions with sugars circulating in the blood, a process that gradually stiffens and cross-links them, a little like the way a rubber band left in a drawer for years grows brittle and loses its stretch even without ever being used. Because these matrix proteins turn over so slowly, the cell's usual quality control systems, built mainly to handle proteins with much shorter lifespans, have limited ability to intervene, and the damage simply accrues. So what emerges, when you put all of this together, isn't a single broken machine, but something more like an interconnected network of maintenance systems, each individually imperfect, each depending on the others for resources and signals, each capable of compensating for a little strain in its neighbors, but not for strain everywhere at once. And it's that interdependence, that quiet reliance of one system on the health of the others, that becomes especially important once we start looking at what happens when cells stop dividing altogether and enter the strange, stubborn state known as senescence. Senescent cells are a good place to pick up this thread, because they capture something essential about how these systems relate to one another, not stacked like independent floors of a building, but wired together like circuits in a single system, where a fault in one place shows up as a flicker somewhere else entirely. A senescent cell is one that has stopped dividing permanently, not dead and not gone, but locked into a kind of enforced retirement, unable to leave the tissue it occupies, while still metabolically active, still drawing on resources, still sending signals out into its surroundings. This state was first described carefully by a biologist named Leonard Hayflick in 1961, when he noticed something quietly odd happening in laboratory dishes of human cells. No matter how well they were fed and tended, ordinary cells would divide only a limited number of times, somewhere around 50, before simply stopping for good. It was a modest observation on its face, made in a lab in Philadelphia with nothing more dramatic than a microscope and a lot of patience, but it overturned a prevailing assumption that cells grown outside the body could go on dividing without end, and it planted the seed for understanding that individual cells carry something like a built-in accounting of their own history. Senescent cells accumulate gradually over decades of ordinary living, and both how quickly they build up and how efficiently the body manages to clear them away again turn out to matter a great deal for how the whole interconnected network holds together. One reason they matter so much is that they interfere with the work of stem cells, the reserve pools of unspecialized cells tucked away in nearly every tissue, ready to divide and differentiate into fresh, specialized cells whenever repair or replacement is needed. Think of stem cells as something like a small standing crew of apprentices kept on hand in a workshop, not doing the daily work themselves, but ready to step in whenever a piece breaks and a replacement is needed. Whether that's a skin cell shed in the shower, a blood cell that has run its course, or the lining of the gut, which turns over completely every few days. With age, this reserve dwindles, both in number and in vigor. The stem cells that remain divide more slowly, produce somewhat less reliable replacements, and become more prone to entering that same senescent standstill themselves, which means the pool of apprentices available to a tissue shrinks precisely as the demand for repair, given everything else going on, tends to grow. This isn't a failure that happens in isolation. The environment surrounding these stem cells, sometimes called their niche, is shaped by neighboring cells, including any senescent ones nearby, whose chemical signals can actively discourage stem cells from doing their job well, nudging them toward dormancy or exhaustion rather than renewal. A workshop with fewer apprentices and more foremen shouting confusing instructions gets less done, and it gets less done in a way that compounds over time. The immune system experiences its own version of this decline, and it happens to be one of the more visible ones, at least to anyone who studies anatomy. Tucked behind the breastbone sits the thymus, a gland whose entire purpose is to train immune cells called T cells to recognize threats without turning against the body's own tissue. The thymus reaches its largest size around puberty, and then, for reasons that remain only partly understood, begins to shrink, replaced gradually by fatty tissue, so that by late adulthood it is a fraction of its earlier size. This process, known as thymic involution, means the immune system produces far fewer new, naive T cells as the decades pass, cells equipped to recognize threats it has never encountered before. What the older immune system has instead is an enormous, well-worn library of memory cells, tuned to fights it has already won, useful against old, familiar enemies but slower to mount a fresh response to something genuinely new. It's a bit like a veteran staff that knows the established procedures inside and out but has stopped hiring, so that when an unfamiliar problem shows up, there's nobody on hand whose whole job is figuring out something nobody has seen before. This changing immune landscape connects directly back to those senescent cells, because part of the immune system's job, under ordinary circumstances, is to find and clear them out, the way a diligent inspector removes machinery that has been tagged out of service so it doesn't keep cluttering the floor. An immune system that is aging in its own right becomes less efficient at this particular task. So, senescent cells that would once have been identified and cleared begin to linger longer, and because they don't simply sit quietly, this creates a feedback loop worth pausing on. Senescent cells release a cocktail of signaling molecules, inflammatory proteins among them, a phenomenon researchers sometimes call the senescence-associated secretory phenotype, which is a technical way of saying that a cell in retirement can still make a great deal of noise. These signals are thought to have started out as a helpful alarm, a way of summoning immune cells to come and clean up damaged tissue, useful in a young body handling an occasional injury. But when senescent cells accumulate for years rather than being promptly cleared, that alarm becomes a low, constant hum rather than an occasional urgent call, contributing to a state researchers have taken to calling inflammaging, a mild, chronic, smoldering inflammation that isn't caused by any infection or injury in particular, but seems to rise gradually with age almost everywhere in the body. It's less like a fire alarm going off for a real fire, and more like a smoke detector with a low battery, chirping intermittently for no clear reason, quietly wearing on the nerves of everyone in the house. There's a further layer to this story that involves something outside the body's own cells altogether, which is the trillions of bacteria and other microorganisms living in the gut, collectively called the microbiome. The idea that these microscopic tenants might have something to do with aging isn't new. The Russian-born biologist Elie Mechnikov, working at the Pasteur Institute in the early 20th century, became fascinated by the idea that certain bacteria in fermented foods might promote long life. And although his specific theories about yogurt and lactobacilli were far simpler than what's understood today, his basic intuition that gut microbes and aging were connected has held up rather well, even as the details have grown considerably more complicated. With age, the community of organisms living in the gut tends to become less diverse, with certain species that had been kept in balance by a younger, more robust ecosystem starting to dominate. This matters partly because a diverse, well-balanced microbiome helps maintain the integrity of the gut lining itself, that thin barrier of cells, only a single layer thick in places, that separates the contents of the intestine from the bloodstream. As this barrier grows less tight with age, a condition sometimes described, a little informally, as a leakier gut, small amounts of bacterial material can slip through into circulation. Material the immune system recognizes as foreign and responds to, adding yet another quiet source of chronic inflammatory signaling to a system already contending with senescent cells and their own noisy secretions. So the picture that emerges isn't one of separate systems each declining along their own private timeline, but something closer to a single conversation happening across many organs at once, where a stem cell running short of resources, an immune system with a shrinking library of fresh responses, a senescent cell refusing to clear out, and a gut lining growing slightly more porous all end up speaking the same chemical language of inflammation, each amplifying what the others are already saying. It raises a natural question, one that becomes harder to avoid the longer you sit with all of this. Whether this whole cascading network runs at roughly the same pace in everyone, or whether some people's internal conversations simply proceed more slowly than others. That question about internal pace turns out to look quite different once you step outside the human body altogether and start comparing species, because nature has already run the experiment a great many times, in a great many directions, and the results are strange enough to make you reconsider almost everything you thought you understood about why bodies wear out at all. Consider the mouse. A typical laboratory mouse, kept warm and fed and free of predators, still tends to live only 2 or 3 years before its systems begin the same cascade described a moment ago. The shrinking stem cell reserves, the senescent cells accumulating in its small organs, the low hum of inflammation rising with age. Now consider the bat. Certain species of bat, weighing barely more than a few grams, roughly the same size as a mouse, sharing a similar metabolic rate, a similarly fast heartbeat, a similarly short gestation, can live 30 or even 40 years. 30 years, in an animal you could hold in the palm of one hand, next to a close relative, by body size, that barely makes it past its second birthday. For a long time, biologists assumed that metabolic rate alone explained aging, the intuitive idea that a faster heartbeat burns through some fixed allotment of life more quickly, the way a candle burning brightly simply runs out of wax sooner. The bat quietly dismantles that theory. Its heart beats just as fast as the mouse's, sometimes faster during flight, and yet its cells seem to tolerate that pace for a decade longer, sometimes many decades longer, without the same accumulation of damage. Part of what makes bats unusual seems to involve their relationship with oxidative stress, that byproduct of cellular metabolism mentioned in earlier parts of this discussion. The free radicals generated as mitochondria convert fuel into usable energy. Flight is metabolically expensive, more expensive, pound for pound, than almost any other form of animal locomotion, which means a flying bat is generating oxidative byproducts at an extraordinary rate every time it takes to the air. And yet, bat cells appear to have evolved unusually effective ways of managing that byproduct, repairing the DNA damage it causes, clearing out damaged proteins before they accumulate into something worse. Some researchers studying bat immune systems have also noticed a dampened inflammatory response compared to other mammals of similar size, a nervous system, in a sense, that doesn't sound the alarm quite so loudly or so often, which, if the inflammatory theories from earlier sections are on the right track, would help explain a great deal about why bat tissue seems to hold up so much longer than the math of body size and heart rate would predict. Then there is the naked mole rat, an animal that resists almost every generalization biologists have tried to build around it. It's a small, wrinkled, nearly hairless rodent, native to underground burrows in parts of East Africa, roughly the size and shape of a mouse, and yet it can live past 30 years in captivity, an almost unheard-of lifespan for a rodent that size. What makes the naked mole rat especially puzzling isn't simply that it lives long, but that for much of its life, it doesn't seem to age in the way most mammals do at all. Its risk of dying doesn't rise steadily with time the way it does in a mouse, or a human, or nearly every other studied mammal. Instead, for a remarkably long stretch of its life, an aging naked mole rat has roughly the same chance of dying in a given year as a young one. Researchers have used the term negligible senescence to describe this pattern, though it's worth being careful with that phrase, because it doesn't mean the animal is immortal, or that nothing in its body changes with time. It simply means that the usual mammalian pattern, that steadily climbing curve of vulnerability that every earlier section of this discussion has been trying to explain, doesn't show up in naked mole rats the way it shows up almost everywhere else, at least not until very late in their unusually long lives. Scientists studying naked mole rat tissue have found several intriguing clues, though the full explanation remains genuinely unsettled. Their cells appear remarkably resistant to becoming cancerous, protected by unusual mechanisms involving a sugar-like substance in their tissue that seems to prevent cells from overcrowding one another, a kind of built-in traffic control that ordinary mammalian cells lack. Their proteins also seem to fold correctly and resist damage more reliably than proteins in other rodents, which matters because misfolded proteins, as discussed earlier, tend to clump together and disrupt cellular function over time, contributing to the kind of cellular clutter that piles up in aging tissue. None of this fully explains negligible senescence, and researchers studying these animals are careful not to overstate what's understood, but the naked mole rat offers a genuinely useful reminder that the steady mammalian decline so often treated as an inevitable law of biology is, in fact, just one strategy among several that evolution has produced, not a fixed rule that every warm-blooded creature must obey. The bowhead whale complicates the picture even further, in a different direction entirely. These are enormous animals, some individuals estimated through careful analysis of amino acids in their eye lenses, and in a few remarkable cases, through old harpoon fragments still embedded in their blubber from 19th century whaling expeditions, to be well over 200 years old, quite possibly the longest-lived mammals on Earth. Unlike the naked mole rat, the bowhead whale is enormous, which on its own offers a partial clue, because across many species, though certainly not all, larger body size tends to correlate with longer lifespan, perhaps because bigger animals generally face fewer predators and can afford, evolutionarily speaking, to invest more resources into long-term tissue maintenance and repair rather than rushing to reproduce before something eats them. Researchers studying bowhead whale genomes have found unusual variants in genes related to DNA repair and to how their cells regulate senescence, though, as with the naked mole rat, the picture remains partial, a collection of intriguing threads rather than a completed explanation. What unites these otherwise very different animals, the long-lived bat, the negligibly senescent mole rat, the 200-year-old whale, is a concept biologists sometimes call the pace of life, or life history strategy, which is really just a formal way of describing the trade-offs every species has had to strike between reproducing quickly and maintaining its body carefully. A mouse, living in a world full of predators and uncertainty, has been shaped by evolution to reproduce early and often, to produce many offspring quickly, because the odds that any individual mouse will survive long enough to benefit from elaborate cellular repair systems are, frankly, not very good. There's little evolutionary payoff in building a body that could last 20 years if something is highly likely to eat you within 2. A naked mole rat, by contrast, living safely underground in a cooperative colony, protected from most predators, has been shaped by a very different set of pressures, ones that reward investing more heavily in maintenance and repair, because there's a real chance that investment will pay off across a much longer lifespan. The bat gains its protection not from burrowing underground but from flight itself, from simply being able to escape most predators entirely, which seems to have freed it, evolutionarily speaking, to invest its energy in long-term cellular upkeep rather than in rapid reproduction. And the whale, protected largely by its sheer size from nearly everything except, in recent history, human hunting, has had the luxury of evolving toward extraordinary longevity as well. This comparative view doesn't hand over a simple formula, a single dial that predators, or body size, or metabolic rate control, that determines how long any given species lives, but it does suggest something worth sitting with quietly for a moment, which is that aging, rather than being some fixed biological ceiling built into every living cell, seems instead to be a dial that evolution has learned to turn differently in different circumstances, faster in some lineages, slower in others, sometimes almost imperceptibly in a handful of unusual cases, depending on what kind of world a given species has found itself needing to survive in. Which brings us, gently, to a question that a person lying quietly in the dark might reasonably start turning over for themselves, which is this: if the dial exists, even if evolution set its rough position long before any of us were born, is there anything a single human being can do, this year, this decade, to nudge it, even slightly, in a kinder direction? The honest answer, the one that doesn't require any exaggeration to be interesting, is yes, and the list of things that genuinely help is shorter and more ordinary than most people expect. It doesn't involve rare berries or laboratories in Switzerland. It involves the things your grandmother probably already told you, though she couldn't have explained why they worked at the level of cells and mitochondria and inflammation, because that science mostly didn't exist yet when she was giving the advice. Regular movement is the closest thing gerontology has to a genuine anti-aging intervention, and not because it burns calories or builds muscle, though it does both of those things too. Exercise seems to reach down into the machinery of the cell itself. It prompts the body to clear out damaged proteins more efficiently, a process called autophagy, which is really just the cell's internal housekeeping system, sweeping up debris before it accumulates into clutter. It improves how mitochondria function, which matters because mitochondria are the tiny power plants inside nearly every cell, and power plants that run poorly tend to leak the kind of reactive molecules that damage everything nearby. People who move regularly, not necessarily athletes, just people who walk briskly most days, who climb stairs, who garden, tend to show slower rates of the biological changes researchers now use to estimate a body's internal age, changes in inflammation markers, in how quickly wounds heal, in how well blood vessels flex and relax. Sleep itself, appropriately enough for a piece of narration meant to accompany it, does something similar. During deep sleep, the brain runs a kind of nightly cleaning cycle, flushing out metabolic waste products, including some of the very proteins implicated in neurodegenerative disease, through channels that open more widely once a person is unconscious than while they're awake. Chronic sleep deprivation, the kind measured in decades rather than one rough week, has been linked to accelerated aging in immune cells specifically, cells that seem to grow old faster in bodies that never get the chance to properly rest. Diet is messier territory, not because nutrition doesn't matter, but because so much of the research is harder to pin down than people would like. Still, a few things hold up reasonably well across many different studies and many different populations. Eating a wide variety of plants, keeping processed and sugary foods to a modest portion of the diet, rather than the majority of it, and avoiding the kind of chronic overeating that keeps the body in a constant state of managing surplus energy. All of these correlate with slower aging by most of the measures scientists currently have. Some research on calorie restriction in animals, mice living meaningfully longer on diets with about 30% fewer calories than they'd normally eat, has generated enormous interest, though the picture in humans is far less dramatic and far more complicated, and nobody sensible is currently recommending that people go hungry in pursuit of extra years. Not smoking belongs on this list too, and it belongs near the top. Tobacco smoke does measurable, well-documented harm to the very systems this narration has spent so much time describing, shortening the protective caps on chromosomes known as telomeres faster than almost anything else scientists have studied, damaging blood vessels, and accelerating the low-grade inflammation that seems to sit underneath so many age-related diseases. If there were a single most effective thing an individual could stop doing to slow their own biological clock, quitting smoking or never starting would be very near the top of that list. Managing blood pressure matters more than it might seem to, sitting quietly in the background of annual checkups. High blood pressure works on blood vessels the way a garden hose left under too much pressure for too many years works on the hose itself, gradually stiffening it, making it more prone to small tears and weak spots. Those small, repeated injuries inside blood vessels are part of what accelerates the kind of vascular aging that eventually shows up as heart disease or stroke, or, more quietly, as a kind of creeping damage to small vessels in the brain. Vaccines, oddly enough, also belong on a list like this, not because they touch the biology of aging directly, but because avoiding serious infections, particularly ones like influenza or pneumonia that hit older immune systems especially hard, prevents the kind of major physiological insult that can knock a body down a level it never quite climbs back up from. An older person who survives a severe bout of pneumonia often emerges from it having lost muscle mass and functional capacity that simply doesn't return, so preventing the infection in the first place turns out to be a form of preserving the very systems this narration has been describing all along. And then there's connection, something that sounds soft, almost sentimental, next to blood pressure numbers and mitochondrial function, but which shows up again and again in long-running studies as one of the more reliable predictors of who ages well and who doesn't. People with strong social ties, close friendships, a sense of belonging somewhere, tend to have lower levels of chronic inflammation and tend to live longer, on average, than people who are chronically isolated. Loneliness appears to act on the body somewhat like a low-grade, ongoing stressor, keeping stress hormone levels elevated in a way that, over years, wears on the same systems that smoking and poor sleep wear on. Modern medicine itself, of course, has already reshaped human aging more than any single lifestyle choice could, mostly by treating the diseases that used to cut lives short before aging itself became the main event, controlling infections with antibiotics, managing diabetes, treating high cholesterol, catching cancers earlier. None of this technically slows the biological aging process in the way scientists measure it in a laboratory, but it changes what a person's later decades actually look like, which is arguably the part that matters most to someone lying awake wondering about their own future. Now, alongside all of this reasonably solid ground, there's a much less certain landscape that deserves to be walked through carefully, without either dismissing it outright or overselling it. Supplements marketed for longevity, things like resveratrol or NAD precursors, have shown intriguing effects in cells in a dish or in short-lived animals like worms and mice, but the evidence in humans remains thin, and dosing that works in a mouse doesn't necessarily translate cleanly into a person. Senolytic drugs, compounds designed to selectively clear out senescent cells, those zombie-like cells that stop dividing but refuse to die and instead sit around releasing inflammatory signals, have shown real promise in animal studies and are now being tested carefully in human trials, but they remain experimental, not something available or advisable for casual use. Rapamycin, a drug originally developed to prevent organ transplant rejection, has extended lifespan in multiple animal studies more reliably than almost anything else tested, which has made it a subject of serious scientific interest, though it also carries real side effects and its long-term safety for healthy people taking it purely for longevity hasn't been established. Metformin, a decades-old diabetes medication, is being studied in a major ongoing trial specifically to see whether it might slow aging broadly, not just manage blood sugar, and the early signals are interesting enough that serious researchers are paying attention, though the results aren't in yet, and interesting isn't the same thing as proven. What ties all of these together, the well-established habits and the genuinely promising but unfinished science alike, is a certain humility about how much remains unknown, a humility worth carrying gently into the next part of this story. So let's sit with that humility for a while, because it's a comfortable place to rest, more comfortable than certainty would be, strangely enough. Every night, whether you're thinking about it or not, your body is doing the same quiet work it has done every night of your life. Skin cells are dividing to replace the ones worn away by the friction of sheets and the drag of a sweater sleeve. The lining of your gut, one of the fastest renewing tissues in the body, is discarding old cells and growing new ones on a cycle measured in days, not years. Your liver is filtering. Your bone marrow is manufacturing fresh blood cells by the millions, and somewhere in a cell in your arm right now, an enzyme is gliding along a strand of DNA, checking it letter by letter for damage, the way a proofreader might run a finger down a page looking for a single misplaced comma. This has never stopped. It didn't stop when you were 20, and it hasn't stopped tonight. Aging isn't the absence of this repair. It's what happens as the repair, ceaseless as it is, gradually loses a step here, a step there, the way a night watchman who has walked the same rounds for 40 years might start taking 2 seconds longer to notice a door left ajar. The door still gets checked. It just takes a little longer, and once in a great while, it gets missed. That image, the watchman rather than the machine breaking down, matters because it changes what we should reasonably hope for. A machine that's breaking down needs replacing, or it needs a repair that restores it to its original state. A watchman who's slowing down needs something different: support, rest, maybe a lighter load, maybe help from a colleague, but not a promise that he'll suddenly move like he did at 25, because that promise would be false, and chasing it would waste effort that could go toward something real. This is roughly where the science of aging stands right now, and it's worth being honest about that, especially at this hour, when honesty tends to land more gently than it does in daylight. Nobody credible in this field is promising reversal, not in the sense of turning back the clock and returning tissues to a youthful state wholesale. What's being discussed, cautiously, provisionally, is something narrower and more useful. Health span, the number of years a person gets to spend not just alive but capable, mobile, clear-headed, free from the kind of illness that steals the texture out of daily life. Health span and lifespan are not the same measurement, and confusing them is one of the easiest ways to misread almost anything written about aging. Lifespan simply counts years, birth to death, a number that ends up on a headstone. Health span asks a quieter, more personal question. Of those years, how many were actually good ones? A person can gain 5 extra years of lifespan and spend every one of them managing 3 chronic conditions from a chair by a window, watching the seasons change through glass. Or a person can gain the same 5 years and spend them walking a dog most mornings, cooking meals they still taste properly, remembering the names of grandchildren without having to search for them. The biology discussed earlier in this narration, the senescent cells being cleared, the mitochondria being kept efficient through movement, the DNA repair pathways being supported by adequate sleep, none of it is really aimed at adding raw years to a lifespan chart. It's aimed at compressing the difficult years at the end, at keeping the watchman effective for longer stretches of the night rather than trying to make him young again. That's a meaningfully different goal, and it's the one worth actually holding on to as you drift toward sleep tonight, because it's realistic in a way that feels sturdy rather than fragile. Immortality, the word itself, tends to sneak into conversations about aging science, even when nobody intends it, partly because headlines love it, and partly because it's a very old human wish, older than any laboratory, older than written language probably, but nothing described across this entire narration points toward it, and it's worth saying that plainly rather than letting the word hover unaddressed. Telomeres shorten, and that shortening serves a protective purpose against runaway cell division, so lengthening them indefinitely isn't obviously safe. It's associated in some contexts with increased cancer risk, an unresolved tension raised earlier and still unresolved now. Senescent cells, for all the trouble they cause when they accumulate, also play real roles in wound healing and in suppressing tumors, so clearing every last one of them isn't necessarily the unambiguous good it might sound like. The mitochondria that power every cell in the body also produce the free radicals that damage those same cells, an old paradox that hasn't gone away just because we've named it. Aging appears to be woven through the same systems that keep a body alive at all, not a separate malfunction bolted on top of healthy function, and that entanglement is precisely why no single fix, however clever, seems likely to unravel it cleanly. This isn't a discouraging thought so much as a clarifying one. It suggests that the honest goal isn't defeating aging as though it were an enemy that could be vanquished once and for all, but living alongside it more skillfully, giving the body's own maintenance systems better conditions to keep working as well as they still can. And there is real, unglamorous comfort in that reframing, more comfort than a miracle cure would offer, if you think about it slowly enough. A promise of reversal is a promise that puts everything on hold, waiting for a treatment that might arrive in 10 years, or might not arrive at all, might work or might not work, might apply to you or might not. A commitment to supporting repair, sleeping enough, moving the body regularly, eating in a way that doesn't overload the systems meant to process fuel, tending to blood pressure and blood sugar over decades rather than moments, staying connected to other people. All of that is available tonight, requires no breakthrough, and works quietly whether or not anyone is watching. It's not exciting. It won't make anyone's pulse race with anticipation. But it's real, and real tends to outlast exciting, in aging science as in most other things. It's worth acknowledging, gently and without alarm, that there is still an enormous amount nobody understands about why aging happens the way it does. Why two people who smoke the same number of cigarettes for the same number of years can end up with such different lungs. Why some centenarians carry gene variants that seem to buffer them against damage that would fell someone else at 60. Why stress seems to accelerate aging in ways that scientists can measure in telomere length and inflammation markers, but still can't fully explain at the level of mechanism. 10 different reasonable explanations were walked through across this narration: wear and tear, programmed decline, cellular senescence, mitochondrial drift, and more. And the honest answer, the one that respects the current state of the science rather than flattening it into something tidier than it is that all of them appear to be true simultaneously, tangled together, each one influencing the others in ways that resist being separated into one clean cause. That tangle might loosen with future research. Parts of it might not. Not because science has failed, but because biology, built over 3.5 billion years of evolution, was never obligated to be simple for our convenience. So here, at the end of a long night's walk through cells and clocks and watchmen, the invitation isn't to solve aging or to worry about it, but simply to notice it as the quiet, constant work it actually is, happening in you right now, in the dark, without needing your permission or your attention, the same steady maintenance your body has performed every single night of your life and will keep performing tonight, one small repair at a time, while you finally let go and rest. Rest well,